Fetal growth restriction is associated with short and long-term effects. In our laboratory, we are particularly interested in studying the mechanisms that lead to metabolic disease. Using a rat surgery model, we ligate the uterine arteries at embryologic day 18, thus reducing blood flow to the developing fetuses. The pups are then born small for gestational age, and the males go on to develop hallmarks of metabolic disease, including insulin resistance, beta cell failure, and obesity. Over the past few years, we have focused our research on early alterations in the pancreatic immune system. As early as embryologic day 19, just one day following surgery, islets have increased transcript levels of immune-related genes favoring the Th2 pathway. We hypothesize it is this early inflammation that leads to the development of type 2 diabetes in the IUGR male offspring. In order to test this hypothesis, we carefully examined the pancreatic immune populations during the neonatal period. We analyzed the immune cells from the islets and non-islet portion of the pancreas separately. It is important to note there are lymph nodes captured in the non-islet cell suspension. The data I will share now are the populations enumerated by flow cytometry. Our ongoing studies include RNA-seq to phenotype the immune populations. In the pancreas of neonatal pups, T cells and macrophages are the dominant immune populations. In just the islets, you'll see that there is an increase in IUGR males of T cells at postnatal day 7. And this is compared to control males in red and also compared to control females and IUGR females. Similarly, macrophages are increased at postnatal day 7 in IUGR males compared to control males, and there is no difference seen in IUGR females. In the non-islets, we see there is also an increase in T cells and macrophages at postnatal day 7 in IUGR males compared to control males, whereas IUGR females do not experience a change compared to control females. Interestingly, all of these populations that are increased at postnatal day 7 return to control levels by postnatal day 14, showing that this is a transient change in immune cell populations. Not shown here, we have used immunohistochemistry to confirm the increase in population and visualize the location of immune cells in both the islets, exocrine pancreas, and lymph nodes. Having identified a transient increase in immune cells at postnatal day 7, we tested if this increase was dependent on interleukin-4. IL-4 is a Th2 cytokine that we've previously published is crucial to the development of type 2 diabetes in male offspring. Interestingly, we found the increase in T cells and macrophages, typically seen following IUGR, was reduced if the animals were given interleukin-4 neutralizing antibody during the first week of life. However, these effects were not attenuated in the non-islet portion of the pancreas. These experiments implicate neonatal pancreatic inflammation in the development of type 2 diabetes following IUGR.